Hello and welcome to today's discussion. So today we shall be looking at the properties of amino acids. We'll begin with the first one. The amino acids are basically amphoteric in nature. Now you do remember from your secondary school chemistry that when you talk about an amphoteric substance, it basically means one that has got both acidic and basic properties. And so the amino acids are amphoteric in nature. Remember what we said? That the amino acid has got a carboxylic group on one end as well as the amino group on the other end. Let me draw you back to the structure of an amino acid so that you can see what we are talking about. Okay, let me just scroll down and show you that particular structure of the amino acid. Okay, there you are. So you can see there's this carboxylic group which we said is acidic in nature, and we have got the amino group that is basic in nature. And so we say the amino acid is one of its properties that is amphoteric. It has got both acidic as well as basic characteristic, and so we refer to it as an amphoteric molecule. The other thing that you also need to know is that the amino acid exists as a zwitterion ion. I don't know how you pronounce it, depends on who taught you English in grade 5. Others say zwitterion, others say zwetonian ion, but I refer to it as a zwitterion ion. So a zwitterion ion is one that has got both charges. And so this comes about because of the fact that the pH of an amino acid basically depends on its environment. So in some cases, you notice that the amino acid tend to be, to have a positive charge, sometimes tends to have a negative charge. And this happens in order to balance the pH of its environment. When exposed to an acidic environment, the amino acid will tend to resist that particular change because it wants to remain at a neutral point, which is what we refer to as an isoelectric point, whose net charge is 0. Okay? Remember, when I've got positive or negative charges, if it's 1 to 1, then you come up with an overall charge of 0. And so the amino acid basically tends to resist that particular change in pH. So depending on its environment, it can have positive or it can have negative charges in trying to balance the pH environment in which it is found. Okay, and so this is the amino acid, and this is a zwitterion ion consisting of both the negative and the positive charges. This one is the neutral illustration of an amino acid. This is an illustration of a zwitterion ion that basically consists of the negative charged ions as well as the positively charged ions. Alright, it also acts as a buffer. I think I mentioned this particular point whilst I was explaining. A buffer in chemistry is a substance that tends to resist change in pH, and so it buffers an environment. And so because of the fact that they do resist that particular change in pH, they tend to buffer, or regulate the pH of a particular environment. Because of them having, either to ionize or release hydrogen ions and so on and so forth. Okay, think of an acidic environment as one that basically consists more of hydrogen ions than the hydroxide ions. Okay, and then you can understand how the buffering occurs. And so in an environment that is acidic, you think of it as having more hydrogen ions. One that is basic has got more hydroxy ions. And so there is either going to be release of hydrogen ions or hydroxy ions in order to balance that particular situation. So depending on where an amino acid is put, there will either be release of hydrogen ions to balance the environment, so that it comes back to that isoelectric point of 0 that we talked about. Okay, so that is how the buffering situation goes. And so this is a neutral zwitterion. This is one in low pH, this is the other one in high pH. And so you can see that in a low pH there's actually more hydrogen ions, whilst because the pH scale runs from 1 up to 14, and so when we say low pH, basically it's between 1 and 5, 6, somewhere there, because between 6 and 7 you get to a neutral pH somewhere there, and then above 8 it becomes a high pH, which is basically a basic one. Okay, from 8 going upwards, you get a basic pH. All right, and so amino acids can be categorized into 2 major groups. All right, and these are referred to as the essential amino acids and the non-essential amino acids. So where does this classification come in? Which ones do we refer to as the essential and which ones do we refer to as the non-essential? So the essential ones are those that the body cannot produce, but instead we get them through our dietary and the things that we consume. And sometimes there are these microorganisms in our body, like the bacteria. I've got a number of essential bacteria in our body that tends to produce some of these essential amino acids. Okay, like for instance, tryptophan is one of those essential amino acids that is produced by a bacteria known as E. coli. E. coli, Escherichia coli, scientifically known. Basically, one of those bacteria found in your intestines is responsible for producing tryptophan. And we shall talk about that process of tryptophan regulation in the T-operon mechanism. All right, and so we are saying the essential ones are those that the body cannot produce. So we get them. How do we get them? Through the things that we consume or the things that we eat. Our diet is made up of those amino acids or proteins that we tend to get through when we consume those foods. The other essential ones, we get them when these other microorganisms produce them. Okay, so in other words, essential, you don't have them in your body cannot make them. All right, the non-essential are those that your body can make. And it's funny how this principle applies in life. Think of it in this way. Because you have got, let me give you this example. Because you have got water every day, you think it's not an essential commodity. But think of someone who is not having water. It becomes very essential because they don't have it, right? And so essential is that thing that your body doesn't have. It cannot produce. It becomes an essential amino acid. And the one that your body can let you produce at any time, a non-essential, because you already have them in abundance. Okay. And so of the 20, they can be divided into 10 essential, 10 non-essential amino acids. Okay. So unlike animals, plants and prokaryotes cannot make all the necessary amino acids like I've mentioned. So you get them through the diet and so on and so forth. Now, the essential and the non-essential amino acids, you need to know the names. So those that are produced by the human body, we said the non-essential, include alanine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. The essential ones include arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and of course valine. Those are the essential amino acids. So the names might sound a little bit weird and sort of difficult for you to remember, and so and talk about the peptide bonds. So these peptide bonds are very essential information of proteins, because remember what we mentioned is that the proteins are made from these small units, or the monomers. The monomers that we refer to as the amino acid. So your question can be that how then do these small units end up forming a protein. Okay, so these small units, remember I asked you to think of a single amino acid as maybe a bead. In the same way, you can join the single beads together and form a chain of beads that people get to wear. Is the same way you should think of the small units that we're referring to as amino acids joined up to form a bigger molecule referring to as a protein. And so the process by which you join those amino acids together, the bond that basically holds them, okay, a bond is a force of attraction. I'm sure you remember ionic bonds, hydrogen bonds, and so on and so forth. So the bond that basically holds these particular amino acids together, for them to join up and form a bigger molecule we are referring to as a protein, are what we refer to as a peptide bond or a peptide linkage. Now the process by which this peptide bond is formed to form a dipeptide is what we refer to as the dehydration reaction. Okay, dehydration synthesis in other books they might call it. Okay, so in dehydration, remember dehydration, D is to remove, hydration, removing hydra coming from water, and so dehydration is the chemical process by which you join things accompanied by the removal of water. So whenever there is a dehydration synthesis taking place, there is always the loss of a water molecule that is involved. And so in this process where a peptide bond is formed, what we are referring to as a dehydration reaction, it is accompanied by a loss of a water molecule. Okay, so the bond is between nitrogen of the amino group and of course carbon of the carboxy group. This forms a peptide bond, and the reaction we are referring to as a dehydration reaction or condensation polymerization. That's what we refer to this particular reaction. Okay. The opposite is the hydration reaction. Okay. And this is the illustration of this formation of a peptide bond. So remember, the amino acid has got the carboxyl group on one end and has got the amino group on one end. So the hydrogen from the amino group bonds with the OH coming from the carboxyl group to form the water molecule that is released, and then a peptide bond is formed. Okay, so this is how a peptide bond is actually formed. Listen.